welcome. I'm Daniela Vasco. If you have met me before, I'm the early career representative for Australia, and we are having our first joint webinar series, joint webinar series with the student early career statistician from New Zealand. And today we have Muskan with us. Muskan, say wave so everyone can see you. She is the new representative for New Zealand, so we see much more of her in our next events of this year and the next year as well. So yes, today's the big news that we have, and we're also organizing a few events for this year, so keep posted. We have a new webinar series coming, and we also have our conference coming up. And I'll start today's workshop with the acknowledgement of country. So in the spirit of reconciliation, the Statistical Society of Australia acknowledges the traditional custodians of country throughout Australia and their connection to land, sea and community. We pay our respects to the elders past and present and extend that respect to all Aboriginal and Torres Strait Islander people today. And today I have the pleasure to have Roy Costilla with us. I'll just check if Roy is already online. Yes, thanks, Roy. And I'll introduce him and get started. So Roy today is going to talk about adventures in statistics and genetics, a brief history of the methods in animal breeding. So in his talk, he will talk about how these two fields, statistics and genetics, changed and evolved in this last century in particular. So in this talk, he will introduce some of the historical problems that intersect these sciences, and he will share his journey in animal breeding and his take on what the future could look like. Roy was born in Peru, and he studied in New Zealand, and he's a statistician and a geneticist, and he research focus on genetics, computational methods, and their applications to animal and plant breeding. So before he joined the agricultural research, he completed his PhD in statistics at Victoria University of Wellington in New Zealand and a postdoc at the University of Queensland, Australia. So welcome, Roy. We're very excited to hear you talking about such an interesting topic. Thank you.
Kia ora. My name is Roy Costilla, and I'm a scientist at Ag Research, and I'm very happy to be here with you this afternoon. I'm just gonna share my screens, and please let me know if you can see them. Can you see that?
Not yet. Not yet.
Oh, yep.
Yes.
Okay, thanks a lot. Thanks again for the opportunity to be here with you all today. As I said before, I'm Roy Costilla. I'm a scientist at the animal genomics team at Ag Research, based in Rockura in Hamilton, in New Zealand. And today I'm just gonna talk a little bit of a brief story of the methods that we use in animal breeding and genetics. I would like to start with the animals that we breed. Here we have a few pictures of animals from different species, going from dairy cattle, from sheep, from deer and buffalo. And you will be thinking, what are they all have in common? The commonality between all these apparently different species is that they are part of different breeding programs, where we use the models from animal breeding that have been developed for over a century now to breed better animals, to select animals with better genetic merit. This in the literature of quantitative genetics is called genetic progress. So it's as a very famous statistician said once, we as statisticians we get to play in everyone's backyard, and this is certainly the case for animal breeding. The outline of my talk, I'm just going to start at the beginning. That I see as the beginning. It could be many beginnings, as we have in different fields. But I'm going to talk about the early days of the animal breeding in terms of the models that we use. Then we'll fast forward to the breakthrough that Henderson in the 50s came about. Then I will introduce the methods that are used nowadays, just to finish up with some conclusions. The early days. So I'm going to start in 1886 with a paper by Francis Galton. This paper it's a landmark paper because it's the one that first used the word regression. And Galton, and this is England in the Victorian times. He started, he and others polymaths from his generation started making quantitative comparisons. In this case, they use height as a trait, and then they start looking at the comparison between the children's and the parents' height. And here in this picture on the right-hand side, we can see two lines, one with the mid-average for the parents, this line with a bigger slope here. And he did exactly the same for the kids, for the children of the parents. And he found that the so-called regression line for the children had a lower slope than the regression line for the parents. And this means that in general the parents, the kids, the children tend to be closer to the mean than the parents are. So in other words, children of taller parents will be shorter, and children of shorter parents will be taller than the parents, because the slope is smaller. And he called this regression towards mediocrity in hereditary stature. What he really meant, it was a regression towards the mean in children's height. So this is a very famous paper that was published in the Journal of Anthropological Institute of Great Britain in 1886. So this is the beginning of the animal breeding models. Then, and I'm fast forwarding again a few decades later. I'm missing Pearson and several others that came after Galton. But the next person I want to focus on is Fisher, Ronald Fisher, that you might be familiar with. In his 1918 paper, he actually introduced the term variance and the analysis of variance. And also he started thinking about testing the natural inheritance theory of Mendel and Darwin. So he started thinking on quantitative ways to test these theories of Mendelian genetics. And in doing so, he also introduced a model that it's used currently, the so-called infinitesimal model that was formalized later on by Bulmer in the 80s. But basically, this infinitesimal model of Fisher postulates that all the regions in the genome will have effect on the trait. So no matter how small, but all the regions will have some effect. and that's why this is called the infinitesimal model, because regardless of whereabouts in the genome is located, all of them will have an effect on a given phenotype. So this is in 1918. Now again, fast forward into Henderson's in 1949. Henderson introduced the estimation of random effects using maximum likelihood. This model is known as the BLAP model, the best linear unbiased prediction model. And this is probably is more part of the statistical literature, because this is when we started studying linear mixed models. This is the first paper that introduced a method to estimate random effects in a linear mixed model. And funny enough, this very famous landmark paper of Henderson wasn't actually a complete paper. You just have it here. It's an abstract of a conference in the US at Cornell. So very interesting. The paper came later on, but everyone uses this citation because the key ideas were portrayed here. And here I want to pause to introduce a little bit of a term that is used in quantitative genetics, which is the breeding value. The breeding value is the estimation of the genetic merit of the animal. So how good the genetics of the animal is, and this term is very important because it's one of the aims of animal breeding to increase the genetic merit of the animals through genetic progress. So in other words, to select animals with better genetics through the time. This is it. That's informal terms in algebra, in the language of equations, if you want. This is the model that Henderson used, also known as animal model in animal breeding. Is nothing but a linear mixed model, where on the left hand side we have our adjusted phenotypes, a vector of adjusted phenotypes. And on the right-hand side, we have, first of all, some fixed effects that we want to control for. And then we have our genetic effects, this term here, A, which are meant to be the additive genetic effects that have some distribution. In this case, assume that they are normally distributed with a given variance. And here I want you to pay attention on the A matrix and the sigma square A, which is the genetic additive variance. This A matrix, it's a very important matrix in quantitative genetics. Is the numerator relationship matrix, which is computed from the relationships between the animals. This matrix is also called kinship or pedigree matrix. Basically, it's an n by n. If we have n animals or n individuals in the sample, it's the expected relationships between all those animals in the sample. So if we have an animal that has a progeny, the progeny will be expected to have 0.5 of the genetics of the sire and the dam. This we're gonna talk a little bit more about this later on. This is not exactly how biology works because of recombination. Those expected relationships are not exactly that, but are something that are close. So for half-zips, that a relationship is expected to be 0.25, for instance. So then the other term on the animal model equation is the random, the residuals, the effect of random residuals. This is just like in any linear model. We have these residuals that have some distribution, which might or not include weights for that residual matrix. But in essence, this is the animal model that Henderson found, postulated the solution for. So that was in 1949. Again, going another leap, 60 years later, how's the animal breeding nowadays? So we go and go into the field and to the farms and get a biological sample. Here it's we have Professor Ben Hayes doing the sample from Angus steers, from hair tail from them. So you get a biological sample, put that together for a whole herd, and send those samples to the lab for sequencing or genotyping, where we have, for instance, in this case, is a Nova 6000 sequencer. And then from that machine, we get the data that will go into the model. In this case, could be SNPs or insertions and deletions. But basically, the format that this DNA molecular data takes, it's a two-stranded a helix of nucleotides. There is four possible nucleotides that we short for ACTE. And the interesting thing about the genome is that the sequence of nucleotides, it's very, very long. How long? In most mammalian species, we're talking about 3 trillion bases of nucleotides. This, of course, includes a lot of repetitive regions. So if we're talking about SNPs, we won't have as many SNPs, but we still have a significant amount. For instance, for cattle, the latest estimations are that there's about 100 million SNPs for different. If we put all together all the different breeds, and for humans, the number is similar to that. So we're talking about 100 million positions in the whole genome that we will need to go into the animal model that we saw before. So it's a lot of data. Commercially, how this sampling is done. So we have this SNP chip arrays that going into the machine, into the sequencer, and these SNP chip arrays will have fixed positions across the genome of that you were using at hand. For instance, for cattle, we have at least a couple of SNP chip arrays. This one on the left is the Illumina bovine SNP 50 that has 50,000 SNPs in the array, and then we have another one that has almost 100,000 SNPs. And SNPs it's short for single nucleotide polymorphism array, which is a difference on only one base on the nucleotide and the sequence. There are other ways of there are other types of molecular variation and markers, but because of scalability and ease of genotyping, the SNPs are the most used molecular marker. And what can we do with those samples that we send to the lab, and then we get the data from? For instance, we can get the estimate the actual genetic diversity in the sample. This is an example for Indian dairy cattle that has actually it's actually a mix. They are crossbred animals. On the left-hand side here, we have two animals from Bos indicus breeds. You have a few of these in Australia as well. If you drive around the country, you'll see these animals with a hump. They have Bos indicus genetics in them. These animals are actually indigenous from India. So in this sample, we have all those animals that are indigenous from Bos indicus here on the left-hand side. And on the right-hand side, we have animals that have foreign genetics, exotic genetics. In this case, because it's dairy cattle, they have genetics of Jersey here on the upper right-hand side, or genetics of Holstein Friesian here on the lower right-hand side. So one of the uses of this SNP chip data is to estimate very accurately the genetic diversity of the animals in the sample. So then in 2001, continuing with the models in this brief history, Theo May Winsen, Ben Hayes, and Mike Goddard published this paper that actually is the first one to use genome-wide molecular markers to predict the breeding values. Before we're talking about 2001, there was no complete genomes of any species. So thanks, and this is again the technological change that changes the or impacts in the models that we use. Thanks to the Human Genome Project in the early 2000s, having whole genomes data was possible. So, but the methodology wasn't ready yet. So this paper is the first time that proposes for the first time the prediction of the genetic merit of the animal using genome-wide markers. And the paper actually does a comparison of least squares, GBLAP, and Bayesian models to predict the genetic merit of the animal. So it's a landmark paper as well. So then after that, the era of genomic selection started, and it hasn't gone yet. This is a technology that we are using nowadays. And how does genomic selection work? Basically, we have a reference population here on the left-hand side, where we have both genotypes and phenotypes. And then we have selection candidates. We have the progeny. We have progeny that we need to choose from. And we use this genotyping or sequencing technology to get markers. With the prediction equation from the reference population, we can identify these effects for the molecular markers. Here are denoted as Ws, so W1, W3, Wn, and so on. And with the genotypes or the sequence data from the selection candidates, we can predict their genetic merit on these selection candidates. So looks very easy, but it's very challenging. Because this model requires very, very, very big reference populations due to the fact that these effects, these W's here, these genomic effects of the molecular markers are very, very, very small. Typically, 2 or 3 or even smaller orders of magnitude than the actual trait. So we have very, very, very small, effects that we need to estimate. So the size of the reference population needs to be considerable. Rule of thumb, we're talking about of thousands, if not more. For instance, for that dairy cattle example that I mentioned before, the reference population we use there, it's over 5,000 animals with both phenotypes and genotypes. In algebraic terms, what is the difference to the model that Henderson proposed in 1949? The only difference it's here in the distribution of the genetic effects. We have our phenotype, we have our fixed effects, we have our genetic additive effects, and a residual. But the distribution of the genetic effects, instead of having the numerator, the pedigree kinship matrix, we replace it by the genomic relationship matrix. And this is where recombination comes in, because as I said before, when we have a kinship, a pedigree, that uses expected relationships between all the animals in the sample. With the genomics, because we know the markers, we can estimate exactly the proportion of the genome that those animals share. So for a progeny of a sire, we no longer need to say that it's going to be 0.5 of the of shared genomic regions. We could estimate that precisely with the data. So that could be 0.45, could be 0.52, depending on the sample at hand. Ben Hayes also has a paper where they estimated that the standard deviation is about 10%. So for cattle. So in other words, the expected relationship between animals, between sires and progeny is 0.5, but it could be something between 40 and 60%. But that's the only difference. Everything else in the model is exactly the same. And this is pretty important, because that means that all the technology that was developed for the BLAB model can be used also for the GBLAB model, just changing that relationship matrix. But the developments didn't stop there. And again, doing another step of 20 years, going to 2023, last year, where there is this paper is Van der Plas and 23, and this is where the Ismostradens name comes from. So this paper is using a model that blends both approaches, the approach from Henderson based on the pedigrees and the approach of Menwiesen based on genomic relationships. This blending of approaches is called single-step GBLAB, because it uses both the pedigree and the genomic information. But this paper in 2023 uses this single-step GBLAB approach in the context of very, very large data sets. How large? This model has 6 traits with over 16 million records, 2.6 genotype animals, 2.6 million genotype animals, and over 26 million animals in the pedigree. So it's a pretty massive model. These authors, they call the approach single-step GTA BLAP. It's an indirect approach that combines both the pedigree and genomic information. This model took about 60 hours to run and almost a terabyte of big RAM. So it's a pretty computationally, it's a pretty big model. Don't try to do this analysis in your laptop. And this is where the brief account of models finishes last year. And now I would like to present some examples in milk yield for several species, for cattle, goats, and sheep, on the kind of things that we use these models for. What do we know about the so-called genetic architecture of milk yield? Milk yield, it's a polygenic trait, which means it's a trait that is not affected just by one single genomic loci or marker, but several, perhaps almost close to the infinitesimal model of Fisher. And turns out that the genetic architecture of milk yield is pretty similar across all mammals, not only livestock species. They tend to have moderate to moderate heritability, which means something between 20 and 30%. These estimates come from cattle, from sheep, and from goats, and they are pretty stable. It's pretty amazing to see such a different species, such a different countries or conditions, and these genetic parameters are stable. The factors that affect the accuracy of genomic selection for milk yield are the size of the reference population, the trait heritability, and the relationship between the reference population and the selection candidates. I did talk a little bit about the first one. The second one, it's a trait that has a higher heritability, will provide higher accuracies of genomic selection given a constant sample size. For the latter one, it's if the target and the reference populations are close genetically, then the accuracy of the prediction will be higher as well. And conversely, if they are very far apart, for instance, if we have a reference population from New Zealand and we try to predict, we have sniff effects, we have effects for those markers, and we try to predict in a population from Australia, the accuracy will be smaller than if we try to do that for a population in New Zealand. This is a plot that shows actually that relationship, and how big do we need the reference populations to be to get a certain accuracy of prediction, given the certain level of heritability. So here, for instance, for a heritability of 0.1, which is the first line here, to get a accuracy of prediction of 0.5, we will need at least about 12,000 animals in our reference population. And you see that this relationship is monotonic but decreasingly it decreases with the number of individuals. So the gains that we get increasing the number of animals in the population are decreasing. Going back to the example in India with the crossbred dairy cattle, this is the genetic parameters that we estimated. We estimated genetic additive variance of 0.42, residual variance of 2.55, and a farm variance of 1.26, which gave us heritability of 0.16. And this is pretty, a little bit lower, but very close to the range that I've gave you before, between 20 and 30% for meal yield. So even in this context of smallholder variance, the genetic parameters were pretty stable. This animal model, either the blab model, the G-blab model, or the single-step blab model, they also allow us to estimate the effect, the actual effect of the molecular markers on the phenotype. There is a one-to-one relationship between the G-blab model under the infinitesimal model assumption to a model that it's focused on the marker genotypes. That's called the SNP-blab model. So when we feed this SNP-blab model, what we're actually doing is running a GWAS, a genome-wide association study, because we're estimating those W's that I showed you earlier, that those SNP effects, those molecular marker effects. So the higher that effect is, the higher the influence of that position in the genome has on the genotype. So for the case of meal yield, this is what it's a Bayesian version of a Manhattan plot. Here on the x-axis we have all the genomic positions in the cattle genome, going from chromosome 1 to genome chromosome 28. And here on the left-hand side, we have a posterior inclusion probability from the Bayesian model. So the higher the probability that the SNP effect is actually different than zero. So the higher this probability, then the more likely the estimated SNP effect is actually higher than zero. So for instance, here we have that in chromosome 4, this SNP near this location is close to a gene that is actually related with milk production and several other traits. Milk protein composition, mastitis resistance, and milk cholesterol content. Likewise with this other gene in chromosome 1. So here we're actually using the model not just to do the prediction of the genetic value, but also using an alternative form of the model to predict the SNP marker effects and run a form of genome-wide association study that could lead us to genes that are associated with this particular phenotype. Going back into the breeding values and the reason for the animal models, one of the very important goals of breeding is to generate genetic progress, to improve the genetic makeup of the population over time. So here we have an example from goats, from Wheeler et al. 2018. These are actually colleagues from Ag Research who work on dairy goats. And we can see that an index based on the breeding values, in this case, is a combination of breeding values for milk yield, fat, and protein. When you look at the trends over time of this index, you see that after from 2013 to 2023, so over 10 years of data, you see that there is a upward slope after the introduction of genomics. So in 2017, this particular company started using genomically informed breeding values based on the GBLAP and single step models, and you see that the genetics of the coming generations are much better than the ones from the past, and this actually can be changed into a monetary values. So this, for the breeders, this is actually the most important thing, because they're at the end of the day, the breeders are interested in having more profitable animals, or animals that will have a better genetics, giving not just one trait, but a number of different traits. So this is quite an awesome success story from the dairy goat industry. That's the end of my talk. I'm just going to finish off giving very briefly some conclusions. The first one is that stats and genetics share similar origins, trace back to the previous to last century, at least to Fisher and Galton. The second one is that the technological advances in the last decades, the availability of SNP chip arrays and fast computers, have provided powerful tools for wider application of these models of animal breeding. The last one is that the methods used in genetic evaluations and the genome-wide association studies for several species around the world these methods are used in applied applied animal breeding. They're not just nice methods that work and have good statistical properties. They are actually applied in the industry, and we all benefit from the genetics gains that these species have, because we are able to select better animals or plants that than the ones from the previous generations. And now I'm just going to start speculating and talk a little bit about the future. I see three things coming, and this is the less serious part of my talk. Please feel free to ask me questions and disagree with me, because this is just my only personal take. I think in the future, the methods that use large amount of multi-omic data and allow more accurate predictions will prevail. And with the new technologies, this also bring even larger amount of data, like all the data that we gather from precision agriculture, from GPS collars, from many other devices. They are sometimes capture real time, so that the amount of data that we're going to be getting is not going to get any smaller. It's going to get much bigger. So the methods that the methods that will stay, it will be methods that will be able to handle that those large amounts of data. And I think they're going to be a lot of indirect prediction and approximate methods. And there, I want to also mention the artificial intelligence, machine learning methods in that arena as well. I think as statisticians, we need to be looking at those. And in the case of animal breeding, interestingly, they haven't performed as well as the GBLAB or single step BLAB models. And this is something still it's still in the research. It's still on the research phase. There is a recent study a couple of years ago that compared over 200 models, and the machine learning models did sometimes as good as the traditional animal breeding models, but there were definitely no clear winner for that. There are a number of reasons, but they are not outperforming the traditional models for animal breeding. So that's the first thing that I can see in the future. The second one is the implementation of these models. I think we're going to see more open source software coming that is available to wider audiences. And here I want to mention three packages: Julia, Python, and R. They uh, don't focus about the order much, but I see that these methods, the software that we will be using in the future, will be more open source, available to different communities. The last one, it's the coming or staying of more polymath people like Galton or Fisher himself. What do I mean by that? I mean that we will need to be specialized, but also have interdisciplinary skills. Not just focus on one specific area, but also knowing several areas and be able to establish the dialogue between the different areas. For instance, in animal breeding, we should be able to as statisticians, talk to biologists, talk to computer scientists, talk to geneticists, and be able to understand the problems that need to be solved. I see more of this coming, and it's interesting because as I mentioned at the beginning of the talk, that's how everything started. That's what the Victorians were thinking of. They were not just thinking; they were even broader. They were philosophers as well, thinking about the problems, and that's how they developed that the models that we're using now. So we need to be a little bit like the fathers, the founding fathers of stats and genetics. With that, I will finish off just saying thank you so much to all the breeders, in that they without them we wouldn't have any models that because they collect the data, they do the hard work in the field. I want to also say thank you to Ken Dotz and Joe McEwan from Ag Research and all my team from Animal Genomics. I also like to say thank you to Ben Hayes from the University of Queensland, Australia, and to Ivy Liu from Victoria University in Wellington, New Zealand. And with that, I thank you.
Thank you, Roy, for the great talk. Very interesting to see how these models are applied in industry, as we saw great results and how it is really improving the field and helping the breeders and also the industry to move forward. I'll open for questions. If anyone has questions, feel free to unmute yourself or type in the chat, or do as you like. But if no one starts, I can start with the question. So in the machine learning field, that's why I was thinking while we were talking, because such large data sets and so computationally demanding, intensive, especially for Bayesian models, as you showed as well, that tend to be more computationally intensive. How do you think the interpretability of these models, like how important is the interpretability of models? Because many machine learning models still are black boxy types. What is your take in that?
No, thank you. Thanks for the question, Daniela. That's a very important and very important question. At the end of the day, what we use in the animal, if we're using the animal breeding models just to be a prediction machine, it doesn't really matter what's inside the prediction machine, right? So but so far, as I said, the machine learning models that are agnostic, they haven't performed better than the traditional models that have more structure, that have either the infinitesimal assumption from Fisher or the Bayesian alternatives to that. They haven't outperformed it, and that we still understanding why.
Yes.
Some hypotheses there are that there's not enough big enough training populations for animal models. So these machine learning models, for instance, in the cases that they have been successful, they have very big amounts of information for the training part, right? Like the image models, for instance, we're talking about very
Yes,
significant. And we don't have that size of the training data sets in animal or plant breeding yet, but we might uh, um, later on. So that's something that's still, but yeah, it could be that once we have trained them enough, then if they outperform the current models, they will be used as well.
Thank you. We have few questions in the chat, one from Christina Feng. So how did you address the issue of collinearity among the SNPs and PCA and Bayesian I1 approach? Linear.
Yeah, so there's two answers to that. The in the paper, the SNP black model actually fits all of the SNPs simultaneously. So then, if there is linkage disequilibrium between different loci, then only the 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 SNPs that have an effect will have an estimate that is significantly statistically significant. So that is the beauty of using the SNP blood model, because you fit all the SNPs all the SNPs simultaneously. When the Bayesian versions of the model, they are even better, because in cases where you have genetic architecture where the where a higher proportion of the variance is explained by certain regions, then that estimation will be even more precise when we have the Bayesian models, base A, base B, or base R. So that's how they deal with that, by using all fitting all the SNPs simultaneously.
Thank you, Roy. Another question from the chat is, can we use gene cloning because it's faster?
What do you mean by gene cloning?
Do you want to unmute yourself as lucky, or you can try to as cloning the animal instead of breeding over time? Is that what she means? I don't know, or he means? Yes,
Yeah,
he said yes.
that actually doesn't that's whether you are able to clone or not animal or plants, that's not up to the statisticians or the geneticists, that's up to the regulation on each country. For instance, in New Zealand, it is prohibited to do to clone biological material unless you are on a facility. As a matter of fact, there is only one place here in our campus in Rakura that is allowed to do genetic modification in the lab, just for research purposes. in other countries it's different. Like in Brazil, for instance, it's not the case. That's not the case. Genetic modification is allowed by law. The European Union has mixed rules as well, but it's something. Yeah, that's something of the future as well. Why? Ideally, if you can do you can generate faster genetic progress if you do genomic selection on embryos, right? So you before they are they are born, you could get a biological sample. I get the genotype them and pick the animals with the best genetic merit. But that is that depends on each country why the regulations are different across the different countries.
Thank you, Roy. There is a comment as well from Abacus Bio. Please say, cloning doesn't get the benefit of the genes. Also, if one clone is susceptible to a lethal disease, then the clones of the whole world would die out. Imagine that happens to which human race can be helped. That's common.
Hi, I have a question. Can you hear me? Yeah. Hi, Roy. I'm also a statistician working in breeding, kind of by chance at the moment. And yeah, I have a question about you talk about the reference population that you train your models on, and I'm wondering if you got any assumptions on this, especially in terms of structure. Because to me, in breeding, it's difficult to have a reference population that looks like a population, as you would call it in population genetics, like, panmixia and all those things. Like, we've got really inbred lines here and there, and they don't cross much. And then, as you said, if you take from one reference population a model and try and apply it to another one, you might get into trouble in terms of accuracy. But also, your reference population, if you want it to be kind of homogeneous, you also run into the problem of having low genetic diversity if it's a long line. So I wonder if you've got any experience with that and how animal breeders deal with that.
Yeah, no, thanks, Johan. That's a great question. And I haven't mentioned when I mentioned the let's say 5,000 animals, I'm making the assumption of certain number of effective population size there. So, for instance, for that number will be more appropriate for dairy cattle. Like, for instance, when you talk about Holsteins, there is about over 100 million Holstein animals in the world, but the effective population size of Holsteins is about 50 or 60.
That's insane. They even got the same patterns, all of them.
Yeah. So, what does it mean? This means that these hundreds of millions of animals have the genetic diversity of 60 unrelated animals. When you compare that to Bos indicus dairy breeds from India, for instance, the effective population sizes are way, way higher than that. We're talking about 700, 800, which means that the genetic diversity that you find in animals of Bos indicus breeds is much, much, much higher. So yeah, it depends a lot on the species. The size of the reference population and the numbers you'll need, it will depend a lot on the species. Yeah.
But if you pool different lines, then you end up with a problem of having not a real reference population.
Yeah. This is exactly the problem we had in India as well, because they are crossbreds.
Yeah.
And not only in India, dairy sheep is another example of that, because the dairy sheep animals, they don't tend to come from one single breed. And actually, the whole the whole sheep industry in New Zealand has that as well, because it's very easy to crossbreed sheep, and there is always new breeds, and that just means that you're increasing the diversity, but also the predictions might suffer from that as well.
Cool. I have another question if no one else does.
Sure, go ahead.
Yeah.
Whereabouts are you? It looks like you're in Wellington.
Yeah, I'm in Wellington.
Yeah.
I'm in Wellington. Yeah.
It
I
just
work
looks
for
windy
Scion.
outside.
Yeah, it is windy, and yeah, I work for Scion, but I'm not in Rotorua.
Yeah,
So I work in tree breeding at the
yeah,
moment. Yeah. My other question was about the methods. So I find that at least in tree breeding, I don't know much about animal breeding, but Bayesian methods are not used much. I believe that computationally they're a bit harder to use. They take longer to fit, but I know there are clearly spaces where they're better, like with non-continuous traits and things like that. But in your experience, because you seem to have both frequentist and Bayesian backgrounds, what's your take on this? And is the problem that we don't have as many tools that are accessible with Bayesian methods, or that computation times are too long, or is it just that REML is so good, everyone goes with that?
it's just a practical problem. If you have the computing resources and the time to wait for the Bayesian models to converge, people will do it. But in practice, even the REML models struggle when we have millions of animals. So the Bayesian models will be even worse than that. However, this is again the current version Bayesian models, but there is also approximations when we talk about Bayesian models. So we shouldn't be closing the doors to that. There is lots of variational approximations and other non-Monte Carlo methods that could also be used here. And that's, I think this is the beautiful thing about the field. It's never ending. It's not going to finish.
Yeah.
Machine learning can come, or new Bayesian methods with approximations will come as well.
Thank you.
Thank you, Roy. A great talk. We are a bit over time, but I don't know if anyone had any questions, if Roy had the time to answer, but yeah, thank you so much for your talk. It's very interesting. Thank you everyone for participating to the discussion as well. In the end, it's great to have questions and get the community with knowing where we are. We can connect in the future. Who knows? We can know each other. So that's the goal of this session as well, to get the community a little bit more involved together. And thank you, Roy, for contributing for that, and thank you everyone for attending. We took a while to get our webinar series going this year, but we have planned webinars for every month of the year, and then in November we have our conference. So I hope to see you all there. And our conference, we have 3 hubs for it. We have in Perth, Hobart, and Christchurch.